Okay, so we've talked about diffusion, which is the movement of molecules from high to low concentration. We talked about osmosis, which is the movement of water from high to low across a membrane. And we talked about facilitated diffusion, which is the movement of molecules from high to low with the help of a membrane protein. The last type of transport is called active transport. So I'm zoomed in on an embedded protein. So I'm still going with the protein theme here, except notice what's happening. So this is the protein. So we have this molecule that needs to be transported, and we have this ATP binding site. ATP binds, and the molecule is moved in this direction. So are molecules moving from high to low? No, they're moving from low to high. They're moving from low to high. That's called active transport. You need energy in order to move against the concentration gradient. So if you're going from high to low, like going down a hill, like you just coast, you don't need any energy. But to go from low to high, to go up the hill or up the concentration gradient, you need energy. Okay, so we've talked about diffusion, osmosis, active transport, and facilitated diffusion. Now, I want you to compare these and think about which of this column here applies to the different types of transport. So if you need energy, we just talked about this, that would be active transport. Diffusion and facilitated diffusion don't need any energy. Okay, molecules move down a concentration gradient. Down meaning from high to low. Which boxes would you check for that? Well, that should be diffusion and facilitated diffusion. Remember, the only difference between these two is that facilitated uses a protein, diffusion does not, but both of them move from high to low. Okay, so if you move up a concentration gradient from low to high, that is called, right, active transport. Okay, what if the type of transport needs a protein? What if the type of transport needs a protein? Well, we know it definitely will be active and facilitated, right? Because that's facilitated means it needs help. It needs those proteins. Okay, if I don't need a protein and I can pass right between the phospholipids, that is called diffusion. Good. Moving ions. Can ions cross by diffusion? Nope, they need some help. So that could be active or facilitated, depending upon what the cell, if the cell wants to go from high to low or low to high. What about large molecules? What about large molecules? Can they pass by diffusion? Nope, they all need help too. So definitely facilitated diffusion, maybe active transport, depending on if the cell needs to go from low to high or high to low. So what about those small nonpolar and also really small polar molecules? They can cross by diffusion. Okay, so I told you that there are proteins in our cell that act as receptors for signals. And so this is really important when we're developing as embryos, because our cells will receive a molecule from our environment, and that will sort of trigger a chain of events that tell that cell to make a protein, make an enzyme, alter some gene expression, and eventually this might tell our cell, hey, this random cell here, you're going to be a brain cell now, and I need you to make some brain cell proteins. So these proteins that are embedded in our cell membranes are really critical for survival. Okay, so we've done diffusion, we've done facilitated transport and active. The last one we haven't talked about is bulk transport. Oh, I want to show you the classic example of active transport called the sodium-potassium pump.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
So the sodium-potassium pump is really important for our nervous system because it's transporting these sodium and potassium ions. Now notice, there's 1, 2, 3 slots for sodium and only 2 slots for potassium, right? So the pump is moving more sodium than potassium across the membrane, which means there's going to be more positive charge here than here because you're moving a lot more sodium to this side and a lot less potassium to this side. Well, that difference in charge is what enables our nervous system to relay signals across the nerves. So active transport, sodium potassium pump, super important for our nervous system. Okay, so the last type of transport we haven't talked about yet is bulk transport. So what if I want to transport a whole bunch of molecules instead of just one at a time? And what if I need to bring in a whole bunch of molecules instead of just one at a time? Well, that's called endocytosis and exocytosis.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use 3 basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
So you don't have to know the difference between pinocytosis and phagocytosis. All I want you to know is that endocytosis means bringing in a bunch of materials, and exocytosis is releasing a bunch of materials. And remember, this is for bulk transport, a bunch of molecules at once instead of one at a time. Okay, so in exocytosis, remember, nothing is in our cells willy-nilly. Everything is encoded in a membrane. and usually they're called vesicles, right? So that vesicle will actually fuse with the outer membrane, and then this part will break open, and that's how it'll release its contents. And alternatively, the membrane will actually wrap around whatever molecules they're interested in bringing in and form a new vesicle. Okay, so that concludes the osmosis lecture. So diffusion, osmosis, facilitated transport, active transport, bulk transport. How are those similar? How are those different? Make sure you can answer that question.